Hello, my name is Fayaz, and I would like to welcome you to this Pathology Congress. I have tried to spell to the best of my ability PathLAKE and beyond, but my capabilities in Dutch stop on this slide. But I'm very pleased to be a part of this event, and I hope that we can have a very good learning experience based on my and presentations by other speakers as well. Before moving on, I'd like to give you an introduction of our Tissue Image Analytics Center, of which I am a part. The Tissue Image Analytics Center at the University of Warwick is aimed at inventing or reinventing pathology with artificial intelligence. I think it's a vision that all of us share. I think we are all aiming towards revolutionizing pathology, digital pathology with artificial intelligence. The goal of our center is to be a cross-domain and a cross-faculty research center that is involved in education and training, us being a part of a university, also petascale data management. But what we really want to do is to develop computational algorithms or AI algorithms that can be used for computational pathology and easing the different workflows that are used in clinic. We'll also be focusing on commercialization, in which we work with different commercial partners to commercialize some of the algorithms that we develop, as well as scanner vendors, so that we are in harmony with various image formats. We want to focus on clinical deployment, which as I said earlier, is our end goal, so that we can help cancer patients as well as pathologists in doing their diagnosis, as well as identifying various prognostic factors involved in cancer and other diseases. If you ask what is the goal of our center, I can summarize it on the slide that you see on your screen. We take in digital pathology data, that being images, as well as other information such as genomics, transcriptomics. We're working on various projects related to multiplexed imaging, as well as spatial transcriptomics, and we want to apply machine learning and pattern recognition on that to gain insights in the behavior of the disease, in the prognosis of patients, as well as identifying different morphologically and pathologically significant constructs within these images and data. So that's the overall goal of our center. I think that most of the work that we do is related to machine learning, and that's why it is important that pathologists and computer scientists like myself are on the same page when we talk about machine learning. So I have prepared a very simple example that can illustrate the concept of most of the stuff that we do. So let's say if we have a very simple task that's got nothing to do with too much with computational pathology, but it's a fun task to understand what actually is machine learning. So let's say we want to separate or classify between pathologists and computer scientists. And for each of our computer scientists or a pathologist, we take an image and we extract certain features from that image. For example, we can extract to what degree is their dressing white, or how many computer screens do they have in front of them. And we collect data so that we know whether a person is a computer scientist or not. And we plot them on this sort of a scatter plot. And on the screen, you can see that there's a red rectangle which shows a person with 4 computer screens in front of them that is wearing black over here. And as the number of computer screens is large, so they score pretty high on this axis, whereas they score pretty low on the whiteness in dressing axis. And they are a computer scientist, which is indicated by this red rectangle. Similarly, if this shows a pathologist who's dressed in white and they're looking into a microscope, they don't have a computer screen in front of them, so we get a plot like this. We gather more data, more computer scientists as well as pathologists, and our goal essentially is to identify whether we can find a line, in this case, that separates all the pathologists in what we call this training data set from all the computer scientists. So it can be this line, but it's not perfect. As you can see, there are some pathologists that should have been below the line, but they are above the line. So this gives us a certain amount of error. We want to move this line so that we end up with a discrimination that is perfect over this training data set. So this line, the second line that I've shown you on the screen, actually gives lower error in comparison to the first one, but it's still not perfect. This line that you see on your screen can now separate out computer scientists from pathologists over the training data set. And that is what we call a classifier. So what we are interested in machine learning is, given some training data, we want to find a cutoff or a line that separates all the examples in one group in the training data set from the other group. However, even doing that does not guarantee that we'll be able to generalize to unseen cases. For example, we have these two. This is David Sneed, who's actually the head of PathLAKE. He's a pathologist, but because he has computer screens in front of them, probably his image is going to be misclassified. Over here is my friend Sean, who's a computer scientist with me in the TIA center. They don't have a computer screen in front of them. They are also going to be misclassified as a pathologist. The idea is not to think too deeply about this particular example, but to understand the main concept, which is we extract some features, we get some training data, and our goal is to identify a very simple discriminator, which is in this case a straight line that allows us to separate examples in one group from examples in the other group. And what we want to do is to use this training data and the classifier to generalize to unseen cases. And we can work out the mathematical equations for these, but if you can make a cut, essentially you can work out its equation, right? So that's not a big deal in this case. We can apply the same concepts to pathology images. We want to collect a bunch of data of pathology images, and what we want to do is we want to, instead of just classifying between cancer and non-cancer, which is more similar to the pathologist versus non-pathologist example that I've just given, we can do even more. We can try to regress the grade of cancer. We can do tumor region segmentation. We can quantify different types of cells. We can do receptor status prediction or mutation status prediction. We can correlate the patterns within this image to survival or drug response and so on. So that is the sort of things that we do in the tissue image analytics center. We develop algorithms for this. As I said earlier, what we do is we make cuts within this space, and that can be done using a very simple model. So think of it, the equation of a line as taking in these two features that I was showing and producing an output of whether a given point is above the line or below the line. So that is like making a cut in this space. And if you stack multiple layers, as I'm going to say, of these pink units that you see, we'll be calling them neurons. If you stack multiple layers of them, you can actually achieve separability in this space that I showed, even when the data is not linearly separable. So if your data cannot be separated by a line, you would need multiple layers to be able to separate these examples of one group from the other group. So if you got some input feature representation, you can stack up multiple transformation layers, and that will help you achieve linear separability. That is the basic concept of deep learning at a very abstract level. And that, if you want to understand that more, I'll put the link of this in the associated resources, or you can go to this website and make a very simple neural network. And the reason I emphasize this sort of a basic or a fundamental, the fundamental concepts of deep learning is I believe pathologists and computer scientists need to be able to connect at this level so that we can do computational pathology in a bit more detail. Now, these machine learning approaches that I've just talked about are not limited only to classification and regression. We can do more, and we have developed algorithms that allow us to now use very minimal annotations, just like simple point clicks that you see on your screen, and classify them into different types of cells that allow us to find the borders and any morphological differences between different types of cells. We've also extended the same approach that we call NuClick onto classifying and segmenting glands in colorectal cancers that you can see on your screen. We've also developed algorithms that allow you to identify, segment, and classify different types of cells in computational pathology images. So over here is a screenshot where we show different types of cells in various different types of cancers using our algorithm called HoverNet, which is actually one of the best performing algorithms in this area. We've also worked on weak supervision techniques. One of the major challenges in computational pathology is actually getting access to labeled data. And in this case, what we want to do is to use minimal annotations in order to generate predictions at the slide level. So in weak supervision, what we do is we have a single label for the whole slide image, whether it is, in this case, MSI positive or MSI negative. And we can develop a machine learning algorithm that actually is able to discriminate between these two different classes. This is an example of a weak supervision framework that gives very high AUC ROCs on a test set. And you can read a bit more about this paper that we call for this MSI classification at the link on your screen. On your screen right now is a computer generated, the largest, to the best of my knowledge at least, the largest synthetic image that we have been able to generate in computational pathology. So this tissue, piece of tissue that you see as an image, does not exist in real. This is computer generated using something called generative adversarial networks. We use these generative adversarial networks in an approach called Saffron to generate these really large 6,000 by 5,000 tiles image, which showed colorectal histology in quite some detail. So as I think the main goal I've been able to convey that we're focusing on computational challenges in computational pathology, and there are a number of challenges that are still out there that we are working towards. One of the biggest one is actually the size of these images is pretty large, and existing resources in GPU and computational memory technology are not there up to the mark. NVIDIA and other partners who are working with us in PathLAKE are doing a very good job, and they're continuously working towards improving this. But at the current time, we can't put a whole image, histopathology image, into the GPU for machine learning. Similarly, whole slide images are multi-resolutional, as pathologists would be aware, and we want to model this in computational, in the computer, in our machine learning algorithms. And there is no fixed way or the best way of doing this. People have tried doing various sorts of things with it, using attention and multi-resolutional imaging, but there is no fix where we were working towards finding those solutions. Similarly, the context, the spatial context, the width of the image that we consider at one time also impacts our decisions from the machine learning algorithm. And as I said earlier, labeling is another big issue. We want to spend the minimum amount of time on getting training data labels, and we want to get away with using the minimum number of samples in training while ensuring that we generalize to these unseen cases. So in this case, there are upcoming approaches on weak supervision and the like. So classical workflows in computational pathology actually work by taking an image, a whole slide image, and patching it up into different patches, and then trying to train a small machine learning model on each one of them. I tend to relate it to this example that you see on your screen. So a computer algorithm only has an idea of what it's currently looking at. And that can be a limitation if spatial context is important in a given scenario. What we've been able to do is to model this problem as graphs, and we've been able to represent a whole slide image in a graph, just like the one that you see. So different regions that within this image show up as nodes in this graph, which are connected to other regions. And this allows us to chug this whole image into the memory at one time in our approach called slide graph that allows us to do classification of various types. So in this case, we have graph-based classification of HER2 positive versus HER2 negative cases, which you can see that HER2 positive has a bit more red, this reddish pattern that corresponds to distinct morphological patterns within the context of our image. It's not great. The AUC ROC scores are not where we would like them, and that is actually the point that I'm trying to highlight, that we are continuously working towards modeling these challenges, problems at our center. There are a lot of opportunities out there, and there are a lot of challenges. I cannot go through all of them in this presentation, but we are working towards, as a field, I think we should work towards ensuring that pathologist recruitment and training involves computational pathology. We want to do reproducible, explainable, and interpretable machine learning. And we want to stay away from over-engineering solutions so that they can be applied very easily in clinical settings, which is the end goal of all of this. We want to do confidence predictions. So when you ask a pathologist whether they are sure or not about something, most of the time they would be pretty sure about whether this particular image has cancer in it or no, but they're also happy to say or consult with other pathologists if they're not sure about something. This is something that we want to build into our algorithms as well. As I said earlier, we want to focus on integrating these novel and upcoming technologies like spatial transcriptomics, in situ sequencing, and multiplexed imaging in our workflows so that we can give a holistic picture and identify novel patterns of disease pathology. And then our goal is to do clinical deployment, but as a field, I think we need to come together to two things at least. One of them is to provide flexible data sharing protocols as well as a stringent validation, and projects like the TCGA, the Big Picture, and PathLAKE that I'm just going to talk about actually provide this sort of framework that allows people to come together and share data. So I'm briefly talking about my affiliation with PathLAKE. PathLAKE stands for Pathology Image Data Lake for Analytics, Knowledge, and Education. We're supported by a £50 million grant from the UK government, which is run by the UK RI. And our goal is to develop a data lake that can be used for analytics, can be used for sharing knowledge and promoting education. So the goal in the PathLAKE is to facilitate clinical adoption of AI in pathology through digitization, which would lead to a well-curated data repository that would promote AI innovation R&D. We would be working towards validation and regulation of different algorithms, commercialization, and of course, clinical deployment, much like the goals of our Tissue Image Analytics Center. What we've been able to do is to put together a data lake model that allows people to submit their data from different centers, which go into a central storage after de-identification and quality control. And on those images, we have developed software that allow people to annotate different constructs within the image so that the type of machine learning we do can be interpretable and we can get good quality controls on that. So we are going to develop these really large repository, which is fair, so that information within it is findable, accessible, interpretable, and reusable. We are also working towards, we have gathered a very large data set of annotations at multiple levels, at the case level, at the whole slide level, at regional levels, as well as cellular level from our pathologist friends. This is an example of some of those annotations. As you can see, we have these very detailed region level annotations as well as annotations at the cell level. We are currently in the process of publishing a paper on a white paper on how any such project, large project, can benefit from our experience of doing these annotations. So we have an annotation guidelines paper coming up. We have developed these tools. One of them is called PathLAKE Gateway that allow us to manage how many different types of annotations have taken place. And then we have a tool called Wasabi that allow us to do those annotations in practice. We have set up image quality control and annotation quality control pipeline that can detect, for example, tissue folding and whether there are any parts of the image that are blurry, which is of course a requirement before we apply different types of machine learning algorithms. We are working towards developing an analytics engine that would allow diagnostic, prognostic, and predictive artificial intelligence on these images. We are also engaged in training and education. We have recently organized a children's competition that we call Beat the Pathologist, in which images were presented to children and they were able to classify them or not. And we were surprised that they did pretty well and almost compared pretty fairly to the pathologists that we had and also the computer algorithms that we were developing. We've also developed master classes for training pathologists as well as computer scientists and forcing them to work together on hackathons for extended periods of time. Our 2021 edition of the master classes actually finished yesterday, and I think you're welcome to join us for the next. So this has been a very condensed introduction to PathLAKE. If there are any questions, I would be happy to address those. You can also visit us on our website, which is given over here. I would like to thank you for giving me the opportunity to join this meeting, and I hope this has been very helpful to our pathologist friends, and I hope we can work towards a world without cancer. Thank you.